advancement, the field of life science is experiencing a data revolution like never before. To delve into transformative potential and challenges posed by this revolution, we are proud to host a symposium on life science data revolution in NABI today. Let us together embark on this transformative journey of the symposium where ideas would converge, innovations flourish, and the future of life science would be shaped by data-driven insights. To navigate us through this landscape of challenges and opportunities, we have today outstanding speakers with us. To begin with the proceedings, may I now invite the speakers and Executive Director NABI to please take seat on the dais, please. May I now request Executive Director Professor Ashwini Parekh to please greet our guest by presenting them a bouquet of flowers. First, Professor Guy Cochrane. Professor Chuck Cook. Professor Saurav Raghuvanshi. Thank you so much. I would now request our Executive Director to please address us on this occasion.
Very good morning to all of you. We have been meeting regularly now. I'm very happy to say that. Day before yesterday, there was a very good reason for us to celebrate the inauguration of the speed breeding facility, which I just showed to my guests. Yesterday, we had the Women's Day celebration. And today we have the science being discussed. So I think it's a really transformative phase for us, but I'm very happy to see that a lot of you could join us this morning, especially the students. I would like to place on record my sincere thanks to our speakers today. Dr. Guy Cochrane, who's going to talk about the global biodata infrastructure and drive for sustainability, which is a very, very important term in terms of our own research at NABI. We always talk about sustainability, and today we're going to see that how biodata, which is generating from biological resources, is going to play a role in a drive for sustainability. Thank you very much, Guy, for joining us at such a short notice. We are indeed very pleased and honored to have you, both of us, you here. Next speaker, we have Dr. Cook, who's going to talk about global coal biodata resources overview and selection process. We are very thankful to you, Dr. Cook, because it was a very short notice. I just spoke to Saurabh a couple of days ago, and I'm so kind of you to have agreed to join us despite your busy schedule. I'm sure getting up early in the morning would not have been easy for you, but thank you very much for joining. I am also joined by our own friend, Dr. Saurav Raghuvanshi, who is not a new name to all of us. He has been visiting us and a good friend of mine, good friend of NABI in terms of the data structure and sharing. I've been taking his help in a number of ways. All of you know about it. But thank you, Saurav, for making it possible. He is the driving force for arranging this half-a-day symposium. And I also want to thank Shrikant Mantri, who is our own faculty, who took the charge. And within few hours, he came up with the program, connected with the speakers, and arranged all the logistics. So I just take a few minutes and tell you this institution work at the Department of Biotechnology, which is under Ministry of Science and Technology. We have 14 different institutions in our country, localized in various regions of the country. Our institution is of prime importance because we are in the heart of the country. We are in Punjab, where the Green Revolution started. So it's not only highly productive in terms of crop, but in terms of the innovative ideas, as you would realize when you interact with my faculty members, all of them work on diverse area of food and nutritional security and secondary bioprocessing from the other institution. The other institution, CIAB, which I just mentioned it to you, has now been declared merged with NABI. So we are one now. which is National Agri-Food Biotechnology and Biomanufacturing Institute. The word biomanufacturing word has been added to reflect upon the expertise which is coming up in a big way. In our budget, which was announced 2 weeks ago, the small budget, there is a special mention of biomanufacturing. So our government is quite supportive of agriculture research and biomanufacturing. As you could witness, the good infrastructure we have, good expertise we have. I'm sure your lecture is going to add more to that collaborative event, and I'm sure we will develop new program, and with your support and guidance, we'll be able to move in that direction as per the mandate of the institution. So once again, I welcome you all for this wonderful symposium. Thank you very much for joining over here. Thank you very much.
Yeah, so thank you, sir. So we begin with the talks, and first talk is mine. So I would request the dignitaries to please take seat in the audience. So thank you everyone for joining us for this symposium today. And today I will be giving a very brief glimpse of what bioinformatic endeavors we are taking up in NABI. So I have titled my short talk as Big Data and Supercomputing for Accelerating Discovery and Engineering. So the data deluge in bioinformatics refers to the overwhelming volume of biological data generated from various high throughput technologies such as NGS, next generation sequencing, microarrays, mass spectrometry, and imaging techniques. So this deluge term underscores the exponential growth in quantity and complexity of biological data being produced, which presents significant challenges in terms of storing this data, management, analysis, and its interpretation. So the rapid advancement of technology has led to a situation where researchers can generate vast amount of genomic, transcriptomic, multiomics data in relatively very short period of time. However, analyzing and extracting meaningful insights from this data requires sophisticated computational algorithms, high performance computing resources, and bioinformatics expertise. So as a first speaker of the day, I'm happy to set the stage for the symposium with these two quotes. So what maths is to physics, what artificial intelligence will be to biology. And biology's emergent properties make it hard to describe mathematically. But as maths is descriptive language for physics, maybe AI will be the language for biology. So these are famous personalities. Demis Hassib is CEO at DeepMind at Google, and Eric Stmitz was former CEO at Google. And the pictorial representation, I'm actually trying to convey that in this current era where almost everyone have actually directly used AI in form of chat GPT or some other large language model. So I consider ourselves to be standing on the shoulder of the giants. And who are these giants? Giants are actually collectively all of us who have produced the content on the internet, where these large language models are actually uh, training based on that particular data. And apart in particularly in life sciences, these giants are specifically NCBI, EMBL, and EBI. So where on day-to-day basis we interact with these databases, where they have actually collected huge amount of biological data. So Government of India recognized the potential in the field of life sciences, and it recognized that supercomputing will be always a preceding step before artificial intelligence comes up. So in 2014, Government of India announced a multi-billion-dollar project of National Supercomputing Mission, where two organizations of government, Center for Development of Advanced Computing and Indian Institute of Science, they both were given responsibility to execute this particular mission. And so approximately a total outlay of 4,500 crore was assigned, and it was to be implemented, as I said, by CDAC and IISc Bangalore. And under this facility, we were fortunate to have one supercomputer at NABI, which is of around 838 petaflops, almost a petaflop machine we have. This mission started implementation of supercomputers all over India in 2019, and we got our Param Smriti supercomputer in 2021. And let me give you a glimpse of the Param Smriti now. Currently, Param Smriti is housed on the ground floor in Nabi. And so mind you, these are almost, to give you a fair aspect size, it's almost 6 to 8 feet height of this particular cabins in which our supercomputing servers are housed and the parallel file system is housed. And we are catering algorithms which are having applications in all these listed areas, of which NABI is currently focusing on computational biology. And there are more than 200 users currently using this facility, which are from neighboring institutes, mainly from ISER, INST, Punjab University, Indian Institute of Information Technology, Allahabad. And they all are using to accelerate their data mining using this facility. So this is an architecture. I won't go in details of this. We have three kind of servers in our cluster. One are CPU-only compute nodes. These take into account all the algorithms which can run on CPUs. Then we have 10 GPU nodes, where NVIDIA V100 GPU cards are actually available. And there are also for genome assembly kind of applications where huge amount of RAM is required. We have high memory compute nodes, which have more than a terabyte of RAM. And in total, we have storage of more than 1 petabyte of parallel file systems storage. So using all this facility, what do we aim to do? We have several research programs, very ambitious research programs at NABI, where we are trying to improve the cereals for nutrition and processing quality. We're trying to improve the fruit for post-harvest and nutrition quality. We also try to do basic biology for crop improvement and food and health improvement. And also underlying computational biology is there as a program for all these research programs. And I'm showing you a very kind of unconventional collaborative network where expertise from different domains, microbiology, biotechnology, engineering, pharmacology, are actually accelerating this agri-food and nutrition innovation engine at NABI. And what multi-omics focus we currently have at NABI to focus on these particular fruit and crops. So annona, lychee, tomato, banana, millets, wheat, rice, Arabidopsis, and we are also focusing on mouse and human genome mining and also microbiome data mining. And specifically with respect to my lab, we are focusing on multi-omics, metagenomics, and also we slowly want to venture into mega science, where from petascale, we want to venture into exascale. And we are currently focusing to do multi-omics analysis using metagenomes, epigenomes. We are also focusing on other proteome kind of data sets. And what we aim to do is we aim to generate tools which are supercomputing enabled, create databases through data mining of publicly available huge amount of data. And we want to draw insights. Specifically, we want to get novel patterns and understanding of the biological system to generate new hypotheses. And as I said, we want to slowly venture into AI and machine learning area. So there, I'll be talking about what kind of machine learning models we have currently actually created. So in 2016, I published one opinion article where actually that time the AI was not that very popular term. And we realized that there are so many articles getting published on day-to-day basis, and it is really difficult to keep track of even your focused area of research. So it is impossible task to even read all these articles by an individual. So we propose that only community efforts will be actually useful to derive very important patterns from such kind of literature. And this will be mundane after AI comes up, like until automated artificial intelligence takes over. So this was in 2016 we proposed, but then although at today AI has enabled a lot of revolution, but then we realized that we cannot ask direct questions to biological literature because the information is not very well structured. One problem which we are trying to address in my lab is we are trying to find novel natural products through data mining of biosynthetic gene clusters from metagenomes. So we want to know which metagenome has which biosynthetic gene cluster, and that biosynthetic gene cluster encodes what kind of bioactivity, like it encodes antiviral, anti-cancer, or antibacterial or CIDR4 activity. So only few examples are there in database, and there is one database which collects such information. So only 2000 entries are there in that database. But now when we want to create a machine learning and deep learning model to predict these bioactivities from biosynthetic gene cluster, not many of such pairs are available. Lot of information is there in literature, but not in the databases which can be used for structural machine learning. So scholars from our group, Hemant and Sunaina, they read almost maybe around 2000 papers to extract all this knowledge and have all this information structured. So we have recently created a data set of these activities. And with respect to the future plans which we have at NABI with respect to computational biology, is we want to democratize and accelerate the computational biology through development of new tools. Our focus currently will be multiomics and microbiome. And we want to build future research highways, mainly by doing big data mining and creating databases for specific purposes. And we want to do scientific computing enrichment to every big data exploration at NABI. So with respect to all the in-house collaboration for scientific computing. And I would like to acknowledge all my team members from our lab and all the collaborators, and also to NABI ICT team for helping to manage the Param Smriti facility, and also funding from DBT and good support from CDAC. I leave you with these quotes, and thank you very much for attention. Due to paucity of time, we will take very few questions immediately after the every speaker's talk. And so you can scan this particular QR code and you will be taken to one particular Google document where you can post your questions. And I will be requesting all the speakers to visit that, and whenever they have time, they would like to actually answer those. So with this, I would like to thank you for your attention, and then we would now proceed for our second talk by Professor Guy Cochrane. And yeah, maybe I invite Guy to the stage for delivering his talk. Yeah. Thank you.
Thanks. Great. Well, thank you very much for the introduction, and thank you for the invitation. So thank you, Executive Director, and to Professor Raghuvanshi for facilitating this and putting this program together. And thank you for all of you for coming along. We've had a brief visit of the facilities here. We've seen some very advanced technology, and we've seen evidence of very rapid and recent growth of this. So it's an exciting place to be. And it's always nice to visit a plant-focused facility, because unlike many other areas of life science or in biomedicine, with plants you see things, you see living things, and that's fantastic as a biologist to be able to witness that. It's still real, and it's still, you know, very much part of our world, very important. So thank you very much for that. So Dr. Mantra's previous presentation really sets the scene fantastically well. Thank you for that. Data are the life sciences. So life science has always been about data, but increasingly it's becoming more data intensive, and the capacity to do things using data in future will simply grow, and facilities such as those here will really enable that. It's true to say that very little can happen without data. And so any scientist asking any research question uses data. They almost certainly reference other people's data, reference data sets. They do meta-analysis. The knowledge that we have is derived from previous data. They stand on the shoulder of the giants that produce the data, as it was said. But also typically people produce new data sets. But to understand those data sets, in almost all cases, you need to be able to use other data sets. So data is the currency of science. And with these advanced data science methods, with AI and so on, this will increasingly be the case. By extension, the databases that exist around the world, the biodata resources, as we call them, that are the databases, the tools, the services, and the visualizations and so on that enable scientists to engage with the content, to engage with the data. These are critically important. And we heard about public databases. So there exist around the world some 3,000 plus different publicly accessible data resources that people use very frequently. They're an essential part of their work. And they're very much an infrastructure. They're a foundation for science. But there are some challenges with that infrastructure, and perhaps not so well known. So just to, I think the room is in the room, we have many researchers who are at different levels actively carrying out research projects. So how many people have accessed a life science related database in the last year? This is the audience participation part. So lots of hands. And so how many have accessed a database within the last month? Again, lots of hands. So it's a regular thing we do. And lots of those databases are within this country, within India, but many of them are the other side of the world. And they exist and they're run and they're very useful, but their sustainability is in question. And that's what the GBC is about, and I'll introduce it. So together, these 3,000 plus data resources exist as a very connected network. We think of it as an ecosystem of different databases and different services that really underpin everything that happens in life science research. They're essential to academia, but they're also essential to industry, to agri-tech, to biotech, and so on. They're highly distributed. They exist in many different parts of the world, and the whole system grows organically, which is unlike most other infrastructures. And so very typically, and I'll have an example of this a bit later on, what happens is a scientist develops a new technology that becomes a platform technology eventually, but they develop a new way of generating data. In order to support their work, they need to build a database and some tools. They use those tools themselves. They offer those tools to their collaborators. But then in many cases, there's a call that comes from somebody who they don't know yet, who they don't collaborate with yet, but they're also using the method. They've read the papers. They want to use the database. They request access. And so some of the biggest databases that we all use around the world have grown from research labs and smaller groups who have built something that's become very, very important. And if you look at infrastructure in other areas of science, if you look at astronomy or you look at high energy physics, in those domains, people get together in advance. They plan from the top down to build a particle accelerator or to build a telescope. And then they spend often many years building it in a very coordinated way, and then the scientists come and they connect into the infrastructure, they run their experiments together. But in the life sciences, just as the data production is very dispersed around the world, so are the databases. And so it's an infrastructure, but it's not organized in the same way as other infrastructures. And that's what brings the challenges that we'll come on to see. So it's already very useful. We use it all the time. It's very important that it remains as it is. But as we see these advances with AI and other big data methods, it will become increasingly important. So the value that will emerge from the data that the world is producing will become increasingly important. But we need to have a data infrastructure that supports this to be able to do that. So I've got a couple of examples. You all have your own examples, and there are many to choose between. What I wanted to do here is choose two examples where publicly available open data resources from around the world have been used for purposes for which the people who actually provided the data in the first place had no idea. And so on the left, we have a project, a study called Serratus, which is a viral discovery project. So it's taking genomics data, raw sequence reads, and looking for signals of all sorts of different viruses, and identified tens of thousands of novel species or putative novel species of virus. So taking huge amounts of existing data and reanalyzing. So these are viruses that will infect plants, infect humans, they're a phage infecting in microbes, and so huge variety there. So fantastic discovery. But the people that originally shared the data sets, they deposited their data into a public database, had no idea that this was 20 years later, this was going to happen. But it's one of the value that emerges from data sharing and putting things together in databases. The second example is on the right-hand side. So this is an AI example, and you're probably, most of you familiar with AlphaFold. So AlphaFold is an AI system for solving one of life science's big challenges. It's been a challenge for a long time. So how do you go from a linear sequence of amino acids in a peptide sequence? How do you go for that computationally into, how do you go from that computationally into a folded 3D structure of a protein? Something that's been a sort of pattern finding, very human process in the past, but AlphaFold really came in and solved it. If you're an out and out hardcore structural biologist, there are some nuances and it's not fully solved, but for the most part, it's practically given us a solution to folding, to predicting protein folding, which has all sorts of applications, as you'll all know. So, so the system is called AlphaFold. There's a database. It now has 200 million predicted folded protein structures, which are fantastically powerful. It can only do that because it had a training set of data that came from a publicly available database, the Protein Data Bank, that has been collected over some 5 decades. Scientists around the world doing their crystallography, submitting the structures, publishing the structures, and all of those become the training set. Without that, AlphaFold wouldn't work. But then how do we have 200 million protein predictions? That's because we have an equivalent sharing of genomics data. So we have genomic sequences. We have predictions of coding regions on those sequences. From that, we can predict an amino acid sequence. From that, we can predict proteins, and then we can feed them into the model. And so to get the 200 million structures, you also need all of the sequence. So and again, another data set that if that hadn't been there, this wouldn't have been possible. So extraordinary things can happen when we share data and when we have a healthy infrastructure of biodata resources. But there are challenges. So the demand is increasing. So people around the world produce more data, denser data, often data of higher quality, but certainly data of higher volume. And that's very productive, but of course it increases the demand on the databases. More data and more difficult to handle. We're already dealing with very large volumes of data. We all love open access. It's fantastic to be able to access science around the world easily without charge. But of course, that it requires databases to serve the literature content, to serve the data behind the literature. So that increases demand as well. Funding organizations are increasingly requiring data management planning as part of grant proposals. So when you write a grant proposal, you're typically asked to say what you're going to do with your data. Again, that puts more burden onto the data resources. And of course, it will almost certainly be the case that we have new platform technologies that emerge over the next few years that give us new data types. They answer new questions. So we'll need new databases. We'll need new data resources to deal with these. If you look at biological imaging at different scales of resolution, then over the last 10 years, it's revolutionized. The methods have been revolutionized, and the data volumes and the importance of the data have grown enormously. So things can change quickly. Things will diversify. But there are sustainability challenges. And so one of the key ones is that the funding for the data resources, so to pay for the databases to run themselves, this has been haphazard and very short term because of the organic way the databases have grown up, the lack of a sort of initial planning. And if you look at this chart on the right, so this is a survey that surveyed a number of databases. They happen to be in Europe. They're called the Elixir Core data resources. So Elixir is a coordinating structure for data resources in Europe. But it's a set of typically quite large quite established generalist databases. And we're looking on the y-axis at the number of full-time equivalents, so the number of staff that were working in those or work in those databases to run them. And then we're looking at, from year to year. And so the study was run in 2018. And the two questions, the first question was, how many staff, so collectively across all of the databases, how many staff are you employing to run your databases? How many curators, how many training people, how many software engineers, how many people does it take to run the operation? And collectively, it's about 300. And that was true 2014, 15, 16, 17. The question was asked in 2018. So the second question was, well, for future years, for how many staff do you have guaranteed funding support? So how many positions can you be assured of to run your database in the next year, 2019, the following year, 2020, year after that, 2021, and so on. And so what you see is that, so this is what in 2018, the managers of those data resources knew. This is what they had guaranteed. And what is not here is what it shows is the uncertainty. And so you see that after 2000, so in 1 year's time, actually there's a little bit, we have a few more. So we've got some money for next year. Things are okay. Following year, well, it's dropped a bit, but it's okay. Next year, it starts to drop some more. But when you get to 2022, so only 4 years in, you've dropped to half the number of people. And so this means that if you're managing a data resource, all of your strategic planning can only be based around a 3-year window. Because surely this is not enough even to run some of these data resources. So this is the state of the funding. Typically, data resources are funded on very short-term programs. And if you can't know what's happening in 5 years and in 10 years, you can't be strategic about procuring the hardware, procuring your cloud services, about planning long-term standards development work, about building the expertise in the staff, building a community of people who value the data resource. So really, this is a very bad place to be in if you're running a data resource, because we know that many of these data resources will be needed in 10 years' time and in 20 years' time. We have to give them an opportunity to plan. So funding has been haphazard, little coordination, and the funding organizations around the world that pay for research programs and pay for the data resources really haven't yet cooperated in a way that can build sustainability and solve issues like this funding issue. It's not managed as an infrastructure, and one of the additional challenges is that because of the way it's grown up so organically, we don't even know where all the parts are. There are databases all around the world. We don't even have a master list of all the databases, so there's work to be done to understand this. So the Global Biodata Coalition comes in to look at this issue. It's a coalition of life science research funding organizations that come together. They recognize the essential nature of the data resources around the world, how important they are to deliver their research programs. They recognize the fragility. They understand the problems of funding and other sustainability issues, and they commit to working together to try to find better ways in which they would cooperate to be able to solve some of these problems and build sustainability. And they do that around a letter of understanding. And at any one time we have, well, so at the moment we have 11 members, and the members of the funding organizations. They're typically national level research funders from different parts of the world. We have a couple of large foundations, but importantly, for the GBC, we have a very broad definition of a research funder. So any organization that is funding a life science research program necessarily depends on data resources. Therefore, they are a potential member of our organization. So it could be an agri-tech company, it could be a pharmaceutical company, it could be a small disease or issue focused charity. All sorts of organizations could become members, and we really do try we would like to diversify this. So we work by really bringing different groups of people together and talking, and it's very much a social exercise. And so we have on the left-hand side a community of research funding organizations, the people who fund research programs. On the right-hand side, we have a pillar of people who run data resources. There's a little bit of history of the research funders providing funding in a way that doesn't quite work for the data resources. Reciprocally, there's a bit of history of the data resources not quite getting what they need and what they feel they want from the funding organizations. And so really GBC has to find the thing in the middle. And a lot of that is about defining what sustainability is. So I mentioned finances. Without finances, you can't run data resources, but with finances alone, you still can't run them well. So it's more than that. So you need to have user community appropriate practice. You need to be delivering quality. You need to have training programs. You need to have sustainable ways of career appropriate ways for scientists and engineers to come into the system. You need to be politically neutral. You need to have separate host organization strategy a little bit from the day-to-day operation of databases and so on. So you need more than just finances, but actually defining it. If you ask a research funding organization what the definition of sustainability is for data resources, they will give you an answer that overlaps with the answer that data resource managers would give you. But there's a very different focus. So a data resource manager typically focuses on, well, sustainability for me is keeping my data resource running and developing tools for this community, that community. For the research funders, the focus is much more about the entirety of the system. What happens when you have, two people in different parts of the world competing to do the same thing? How do you get them to work together to reduce a bit of the redundancy? How do you make sure you don't have one country running the really critical database for our country? There are all sorts of issues that relate to life cycle management. So how do you know when actually it's time for this database? It has been very important, but now we need to smoothly deprecate. We need to keep the data, but we don't need the active services. So how do you identify data resources in that state? How do you know when a new one needs to emerge? We need to have a new data type, really important. We need to seed a new data resource. So part of our work is to define what sustainability is, such that we can move towards it. We need to understand the landscape. As I said, we don't know where everything is, and really we need to build cooperative models, mechanisms for research funding organizations to work together more to support data resources. So the GBC has two stated aims. First is to be a forum for the funders of data resources to better coordinate, to share approaches for efficient management and growth, and to share strategies around that. And the second aim is to stabilize and ensure sustainable financial support for the global biodata infrastructure, with a particular focus on a prioritized set of global core biodata resources. And I'll come back to talk more about the global core. So that translates into a whole strategy. So everything that we do is purposefully very open. If you go to our website, well, the link's down here specifically, but go to the website, all the things I'm covering are described in some way there. This is our strategy and program of work. So if you want to understand which bits relate directly to you and how you might get involved, this is a good chance to get some more detail. Essentially, we have a set of things over the next few years that we will deliver, and a set of ways of measuring our success. And one of our priorities is the global core that Chuck will say more about. Another is broadening and diversifying the membership, so we really have a good coverage of the organizations that need to know and need to care about this. So when we go to an organization to suggest that they might become involved with GBC, of course, they need to know why. One of the things that the organizations that have joined really value is the fact that they have a fairly neutral forum in which they can discuss. There aren't so many opportunities for funding organizations in, say, India to talk to funding organizations in, say, North America. So it's very useful as a knowledge exchange. An example of that, we have a number of working groups that are appointed by the members. One of these is on open data strategies. So there's a lot of talk and a lot of discussion about how you manage nationally open data policy and how you get open data policy to work. Some organizations have taken different routes. Some are more advanced in one area, others are more advanced in another. So the sharing of expertise and knowledge has been really important for people. They have discussed quite extensively this lifecycle management issue I mentioned for data resources, and they've discussed how to work or how to build open data policy that is appropriate for better cooperation around sustainability. But perhaps in the longer run, this is about an opportunity to steer things. So an opportunity to sit at the table and be part of the conversation. So to help to design the solution, the sustainability solution that will be applied around the world. It's an opportunity to steer the investments of others. So it's an opportunity. So this will be an investment by different countries into different non-country funding organizations. It will be an investment into a funding model, a model of cooperation. It's very important to be part of the design of that, and that will help to prioritize how other people invest their funds. So there's a leverage effect, but also stating what the priorities are. So not all, if it's country level, not all countries are interested in exactly the same things, and there are some things that are priorities for one country, but most countries don't care about. So really having those on the table so that whatever is built is compatible is really important. There is of course, a more negative thing that if we don't solve this problem, if people don't get together to solve this problem of data resources, then they will disappear or they will reduce their functionality. And if you imagine the NCBI resources, the EBI resources, SIB resources, the protein data bank not being available, which is a longer term risk, then these will have some serious consequences to science. So in India in particular, there is already a lot of activity in providing data resources and consuming data resources. We saw the hands earlier. There's a lot of use of databases around the world. The numbers at the top here are from a study. It's shown on the right. It's a study that just looked at one data type within a particular time window. And it was text mining based, and as we all know, not all texts are open for mining. It was looking for accessions of sequence records, and many people don't write their papers, and even if the text is open, they don't write their papers in a way that cite the records where you can actually find them. So this is it's a huge undersampling, but so the absolute numbers are very small, but the relatively they're quite meaningful. And so taking this approach, so just for sequence data, looking within the time window, 1,277 scientists working in India, so in Indian institutions, they were citing sequence records in this case from 100 different countries. So from studies or from samples that have been sequenced in 100 different countries. So there's an access from Indian scientists to data that are produced on samples from elsewhere. The second point is that Indian scientists use, for their own data, they use global data resources. And so there are 2,463 scientists in the study that cited sequence records that were from, perhaps their own labs, but from Indian laboratories. So these were Indian data sets, but they were accessing them through international databases, so accessing them through databases that are not in India or not yet in India. And then, of course, India is a great provider of data to the world. So 3,559 scientists working in 161 countries outside India are accessing data that relate to samples in India. So this is the numbers are bigger in India than in other parts of the world, but this is a very common pattern that if you're involved in providing data from samples in your country to scientists outside the country, you're also consuming, and you're also a user of international databases. So it's a very international, very connected world already, and we know this anecdotally, but we have some numbers to show the patterns. So we had a very enjoyable week last week at the IBDC. Who were hosting a biocuration meeting, and we learned a lot about the data resources that are operating there, and some new ones that were launched last week. These are India has a rich history of providing databases and providing services to people outside India, but these were nicely condensed into a package, and we learned a lot about them. And it's a fantastic initiative and going very quickly and very successful. So these given as examples. We've built, I'll come on to talk about how we've done this, but we built an inventory of databases around the world. That's where we get the 3,000 number from. We can identify a funding source in India in 31 of those databases. And so these are largely not in India, these databases. And 253 authors who have described those databases are working have at least one affiliation in an Indian organization. So there's a huge contribution for people into running this infrastructure already. This is not a new thing. This is about understanding how it works and coordinating it just sufficiently to assure sustainability. I won't go into details on this. Essentially, for organizations joining, there's an opportunity to be an observer for 2 years, and then the decision making happens when full membership is achieved. So far I've spoken mainly about what we do with the funding organization. So this is now moving over to the community of people who provide and run the data resources. We have to understand the global infrastructure to be able to understand its vulnerabilities. We understand it as an ecosystem, and in an ecosystem you have keystone species. And so what we have in the biodata resource ecosystem is analogous to that. You have a comparatively small number of data resources that are particularly well connected. So they're a deposition database, so a repository of data that's providing data to many different databases, perhaps, or they're a knowledge base that is a compendium of knowledge about a particular type of biological molecule. And the thinking is that if those many things are important in this ecosystem, but these keystone data resources, if we can identify them, have a particular importance because they're so connected that if they're damaged, if they're not sustained, then many of the more things around them will suffer. So outside those keystones, we imagine a long tail. So the very end of that long tail are probably some very, very specialist databases that are critically important to a few people in a particular domain, and then in between you have less specialism and more generalism. So we sort of split our understanding into these two parts. The first part is the keystone resources, and we call these the global core biodata resources, and we run a selection process, an application-based process for people who run typically the big generalist long-established databases that have an international exposure. We invite them to submit, and they go through an evaluation process against a defined set of criteria, and there's an external expert review. Chuck will describe that in a lot more detail, so I won't say much more about that. But essentially, we've run that process twice. In 2022 and 2023, and we now have 52 global core biodata resources. So in there, you will see the genome browsers, the structured databases, sequence databases, and many of the things you already know about and you use on a regular basis. Typically quite generalist, very heavily used, typically quite heavily supported, and typically quite big operations. But what this does is it gives us a great community of people who are working, whose job it is to run these databases, who understand the challenges, they understand the funding issues, they understand the other sustainability issues. And when we do, when the cooperative mechanisms between funders emerge, this is the group that will be able to evaluate them and help to tune that we're building something that's really realistic. So we have a group that the managers of these resources come together in what we call the GCBR forum. Many representatives we try to meet every couple of months. We've had some interesting discussions already. One of the outcomes has been the establishment of another working group to work on what we call the sustainability indicators. So this is really trying to formalize our understanding of what sustainability is. So what are the key features of a sustainable data resource that are desirable? That's a useful evaluation tool, but it's a useful communication tool to communicate this to the funding organizations. It's also a useful tool to plan strategy and development if one has a set of targets to reach for a data resource. As I said, Chuck will say more about the process itself, so I'll move on from there. So for the long tail, for everything else, we need to do other things. We can't have a selection process that we won't be able to reach the people. They won't hear about it necessarily. So we did a literature-based study, the inventory study. We carried it out over the last year or so, and the papers just come out in PLOS One, link there. And if you're interested in reproducibility and open software and open data, all of this is very carefully captured in a second paper that's been submitted, but it's available as a preprint. We really wanted to produce a process that could be rerun by anybody, and we couldn't find, people perhaps have identified these things, but we couldn't find a standard protocol for the combination of computational workflows, parameterizations, data sets at input that would a protocol for describing and depositing all the code and all the components in a particular place. So we asked an expert to come in and review it to see if they could, before we submitted, to see if they could reproduce it. And they came up, sure enough, they came up with some suggestions and some changes. So we really are working towards what we hope is a very reusable methodology. But anyway, we deployed it ourselves. We went to the basis is that if you are running a data resource and you want people to come and use it, then you write a paper to describe it and to invite people to come and use it. Not every data resource does that, but many do, and it's in many areas it's quite a tradition. So that was a starting point. So it's a combined search matching process with some natural language processing, and we end up with a confident set of publications, of literature publications describing databases where we know, or we're very confident that the paper is actually describing how to use a database and the presentation of a database, rather than a paper that's just using a database in some way. So we end up with 3,112. They're very diverse. They're in different parts of the world. We have funding information. We can go back and pull out different bits of information. So we care about who's funding them. And for 55% of them, we have some information about funders. Turns out there are 1,800 different funders funding these databases. That's a huge number. Actually, some of those are very, very small, very local university departments and so on. So perhaps they're not quite the funders we think of, but that gives us access to more people for whom this is a really important issue. It also gives us access to the people who run the databases, who write the papers, a great community to be able to try to engage to help to build sustainability in this area as well. So I'll quickly go through. So the data resources are found in, they are funded by organizations in many different places, but the map is not even. Some parts of the world don't light up. Some parts of the world light up more intensely than others. And that, I think, is an issue. Science is happening in a very distributed way around the world. Data are being produced, and there's a need to access data. So that, to me, pushes for a more distributed data infrastructure. So more funders need to be involved. And if you look at where the managers of the databases are working, so typically this is where the databases are hosted, you have the same thing. Lots of parts of the world light up, but it's not quite so even, and there are some gaps. So I think there is actually an important piece of work here to try to build this in a way that we balance where things are happening and where science is happening, where science is needed. We balance that with an infrastructure that can match. Here's an example from. So we heard about this example last week from Professor Mukesh Jain at the BRIC-NABI meeting, and so this is chickpea, and there aren't so many people, or weren't so many people in the world working on chickpea genomics and transcriptomics at the time. So he produced the CTDB, which was a transcriptome database with data and a set of functions that became useful for a community of people. But he's a researcher. He produced that as part of his own work, made it available to collaborators and made it open, which is a great thing to do. But he's not there to provide infrastructure. So he moved on to different things, and that had to close. And then there are people who write and say, well, actually, where is it? Can you restore it? We still need it. There's a good ending to the story, because on the right-hand side is a new thing, which is an expression atlas, which is richer and deeper with new data. So it's actually doing all the things and more than the previous version, but people don't necessarily know about it. But so this is a success story, but it does illustrate that, there's a real sensitivity here. The community depends upon things. If they're produced by a research group and the research group has to change its focus, this is a real problem. If there's no plan for sustaining them further, for picking them up and running them somewhere else, or for seeding their development in a place that can run them for longer. So and there are many examples of this kind of thing. So a lot of our time at GBC over the last couple of years has been spent on specific strategic discussions with working groups. I mentioned open data. I'm going to mention the sustainability working group now. And this is really working with groups of people who are experts on the funding side, and they brought in people on the data management side, and institutions that run sets of data resources as well. So there was a consultation. It closed early this year, but I'm showing it, so you can't, sadly, there's no opportunity at this point to contribute to the consultation, but I'm showing it because the consultation papers are really rich and useful, and already give an indication of where this is heading. So our work now is to pull in all of the different feedback we got from the consultation and to turn the consultation papers into white papers. But we already know a good deal about the kind of content that's going to be in there. So do look forward to the white papers. We hope for sustainability the middle of this year, open data later this year. But certainly go and look at the consultation papers in the short term. These really give a lot of the thinking from the funder perspective, and a lot of the strategizing from the funder perspective as well. And so in particular, I'll focus on two things. One is a set of principles and models that define how we should approach sustainability. And I won't go through all of the principles, but the first one, for example, is that those who fund life science research share responsibility for data resources. It doesn't mean that they all have to pay for them, but it means that they have a stake in them, so they have to help to find the solution. It talks about funders of all types. Importantly, number 5, it mustn't require, whatever solution we have, it shouldn't require payment at the point of use. We don't want to start people, we don't want people to have to pay for access to these public databases. It needs appropriate governance structures, and the data needs to be open, and there needs to be sustainability strategy in the data resources. So a set of principles that will guide how we begin to build these models of cooperation. And then the second thing, already suggesting some models, and these are probably not so visible from the back, but these are very high level, but these are some of the things that the funders might work together on. So the first one is sort of obvious, that you have lots of funders, different parts of the world, they get together, they contribute to a fund, central fund, and there's some process that fairly distributes the fund to the data resources that need to be supported through some mechanism that we'd have to build. So that works already for some of the data resources, but it very soon, and it might be part of the solution, but it very soon falls apart. And it falls apart particularly because national funders are uncomfortable investing in activities that are happening exclusively in other parts of the world. And there are very few, well, there are no countries, I think, that have no limits on that. So countries do not politically want to invest outside, hugely outside their country. They would much rather invest within their own country. And so the second model addresses that, and it says, well, let's not try to move the money so much, the investment so much. Let's move the activity in the data resources. So if you have data resources more distributed, where you have activities in country A, in country B, in country C, and so on, then everyone is investing locally. They're setting up buildings, machines, capacity, expertise. They're building their own sector. Politically, it's attractive, it's economic activity, but scientifically, it's attractive because you're bringing all this expertise into the management of the data resources. You're bringing the domains of science that are understood in different parts of the world together into managing and curating the data. So there's a great scientific opportunity here, but the model needs to be that the data resources would spread out and there'll be more engagement and there'll be new nodes and new partners who are actually helping to build and benefit from the data resource in more parts of the world. Ultimately, and there's some variations on that, but ultimately there won't be a single solution to this, but there'll be a mixture of some of these different ideas. But certainly in the paper, you can already read about, in the consultation paper, already read about some of the thinking that's coming from the funders there. So just finally, to come back to this issue of openness, we engage a lot with some of the legal negotiations that are happening about data sharing, genomics data sharing in the context of access and benefit sharing. Again, we think we can help to build sustainability models that actually support a more global, more equitable approach to running the infrastructure itself. We want to be more inclusive. We need to be more inclusive. And we also need to be inclusive in the life sciences of other disciplines. And plant science is a good example where you can't just study plants alone. You need to access environmental data sets. You need to know the conditions under which the plants grow. You need to know the historical conditions under which the plants evolve. So you need access. You need to run queries on the data that go beyond the life sciences. And similarly, people working on climate change, people working on environmental sciences need to be able to come in and understand things about the biology of the systems they're working in. So we need to provide this. So GBC is about the life sciences, but it has to work with lots of organizations that are involved in standardization and interface development and connections and similar activities that go beyond. And so in plant sciences, a lot of this is environment, but in medicine, biomedical work, a lot of it's with health data and so on. A good part of our work is interacting with groups that are doing similar things, related things in different domains. So with that, I invite you to follow us in different ways. Do go and have a look at the website. Delighted to answer some questions in the document or to receive any comments and hopefully meet some of you as well. So thank you very much for your attention.
Thank you so much, Dr. Guy. Maybe we can have one live question if somebody has. Yeah, if not, then we thank you so much. And our next speaker is Dr. Chuck Cook, who is also a program manager at Global Biodata Coalition.
Uh, Hello, thank you for the organizers for inviting me. Today, as Guy just mentioned, we had developed a process for identifying those keystone species, in other words, the very important data resources in the greater infrastructure of data resources around the world. And I'm just going to describe fairly quickly how we went about that and what is the process that we developed. There's a huge amount of detail in this. I don't expect anyone to remember any of the detail. Just remember the concept that there is a process, there is some detail, and that we've developed it, and that we, the Global Biodata Coalition, would be happy to help develop similar processes, for instance, in India, to identify important data resources within India. So as Guy mentioned, we've named these resources as global core biodata resources. There's a list of criteria that or descriptions of what they are. They're free, they're open access, they're used extensively. In short, they're important for a very large number of researchers around the world. So how did we take this list of qualities and turn it into and identify resources that had them? This is just an image from our website showing the first 4 of the current global core biodata resources. There is, of course, a list on our website with information about them, a short description, and the funders, where they're located. So when you're setting up a selection process for something like this, it's really important to put a lot of time in at the beginning. What is the purpose of the selection process? What do you want the end to be? And in this case, our end product was, what are important resources around the world? And then we wanted to design the selection process to meet that purpose. And we wanted to ensure that it was a fair and open process. In other words, there was no nothing going on behind the scenes. There was no pre-selection of the resources. We did not know what the result would be. There were no political, machinations to ensure that some resources got named and some didn't. The other important concept here is that the resources themselves had to believe in the process, and that's why it was open. And the funders who are giving the Global Biodata Coalition money to run this had to believe that we were doing it fairly and openly. So we have published a paper about this. It's here on Zenodo. There's a link down there. We'll send the slides around. And we hope that it's actually pretty easy to find in Google. All of the details and all the slides that I'm going to show are in this paper. So I haven't put any more links in this presentation, but they're all linked. Everything links back to this paper. So what we did was we developed a whole series of indicators, and I'll go through them in a minute. But the indicators are both quantitative and qualitative. You cannot use only quantitative indicators to identify, in this case, these resources, because they're so varied. You can't pick any single number that will be appropriate to all of them. So there are qualitative indicators as well. And there's the quote here, not everything that can be counted counts, and not everything that counts can be counted. So we can get a lot of numbers from these resources, but they don't necessarily mean anything. They don't necessarily give any indication of their importance globally. The indicators are used to inform the peer review process. So what we mean here is that there's no just number at the end. We can't put a number on these things. We don't just put a number on the on the applications and align them up. It doesn't work like that. The reviewers, the peer reviewers, go over each one and make a judgment on them. Okay. Now I'm going to go through these indicators. There's a long list of them. I don't expect you to read all of them. I'm not going to read all of them. But there are categories, and the categories are, we think, interesting. So one, of course, is scientific focus and quality. We're doing science. So of course, the scientific aspect is important. We put that first. Some of the indicators are not something we use to make any of the selection. They're just for information. For instance, the first one, deposition database or knowledge base. What kind of database is this? This does not affect the selection in any way. It's just, we just need to know this information for reporting purposes. Scope, what does the database do? How global is it? How many people work for it? Now, again, how many people work for it is not something we would judge it by, but it's something we're interested in knowing. Community, who uses it? How many people are citing it in the literature? How many connections are there to other data resources? This is a really important piece of information. It's also very difficult to understand. Now, as Guy mentioned, there are over thousands of data resources around the world. Most of them are exchanging information with other data resources. So in addition to you, the researchers getting information, they get information from each other. So, but some of them get information from hundreds of other data resources, and some of them may only be connected to one or two. So that's an interesting piece of information that we would like to know. We don't actually collect a number on that because it's very difficult, even for the resources themselves, to find that number. But we can get some idea of it in a qualitative fashion. Then there are some indicators that relate to the technical services of the data resources. Are there identifiers or accession numbers or DOIs or something associated with the data resource? What kind of data volumes are there? Is there technical performance? Is it available all the time? Does it use the appropriate standards for storing and exchanging information? Is there good documentation of how it's curated? Where do the data come from? Is there a quality assurance pipeline or method? Is the data available to everyone? And how much user support is there? Is there a user help desk? Is it possible for user feedback to have any positive effect? Does the data resource run any training? Is there a communications person who does Twitter or whatever? What language is it available in? Now, in our case, we said that the data resources had to be available in English, but some of them are also available in other languages that are local to wherever they are cited. How are they funded, and how long are they funded? Have they been funded for? Now, as Guy just pointed out, most data resources don't have secure funding for the long-term future. In fact, that's why Global Biodata Coalition was created. So we didn't actually ask them how much funding they had going into the future, because almost all of them would have said, this is bad news. We're always struggling to get funding. So we didn't ask them that question. What we asked them was, how long have you been going? If you've been going for 20 years, it's probably a good indication that you can keep going in the future, even if you don't really know where the money's going to come from right now. Are there provisions for data preservation? Are the data open and available? Is there a privacy or an ethics policy, if that's appropriate, which it usually is? Privacy is with regard to both the users and the data itself, if the data needs privacy. And this category, impact stories, is quite important and uniformly the most difficult for one for the resources to answer. How do they accelerate science? So when we ask them this question, it's basically a text essay. It's like an essay on a final exam. We ask them to write something about how their resource is accelerating science, and we asked them to write a counterfactual. And what that means is, if this data resource disappeared, if they lost all their funding and went away, how would that impact science and researchers around the world? And again, of course, you don't really know the answer to that, but you can speculate if you understand who is using your data resources. So these are the hardest to write. These are the hardest for the reviewers to evaluate, but they're actually quite important in the evaluation process. Okay, so the next few slides, I'm going to just go over how we did the review process. So it's operationally similar to those used by funding agencies. Some of you, perhaps many of you, have applied for grants from a funding agency, presumably in India, but possibly elsewhere. And you submit an application. There are reviewers. They are hopefully independent reviewers. They make a recommendation to the funding agency, and the funding agency then tallies all the recommendations for all the different grants and awards money to the ones that are at the top of the list. So the reviewers that we had for this were first our scientific advisory committee, which is about 9 people. And then experts in biodata resource management, including Dr. Rakavanshi, who's speaking next. He was one of our reviewers. And critically, the reviewers didn't include anyone who works for GBC. We were not involved in evaluating the applications in any way. Our function was only to manage the process. So then the reviewers sign a confidentiality statement and a conflict of interest declaration. So in other words, if someone is applying who's the brother of one of the reviewers, then that they won't review that particular application. Or even who is in the same research institute or has co-authored papers with them. Now, for this exercise, we did a 2-stage process rather than a single process. And the purpose of this was to ensure that the first stage was much lighter. It was easier for the applicants to apply. And it was easier for the reviewers to evaluate the first stage applications, which we called expressions of interest. And this was because the actual full application is fairly time consuming, and we didn't want to waste the time of either the applicants or the reviewers for those that would not make it through. So the review committee then decides after the expressions of interest, which applicants were invited to make a full review. And in the full review, there were then very detailed and a much longer form application asking for more numbers and more written responses. So the expressions of interest was divided into 3 sections. The first section 1 was not used for evaluation. It was just information. Describe your biodata source. What kind is it? And it's a member of a consortium. In other words, a group of resources that are collaborating. Tell us about it. This had nothing to do with the evaluation. It's just we need to know this information so that we can help understand the global infrastructure that Guy referred to previously in the last talk. Okay, the second set of questions we asked were eligibility criteria. And I won't detail them, but they had to do with how mature is the resource? Has it been around for a long enough period of time? We didn't allow very new resources to apply. So for instance, if you founded your resource just a year ago, it was not yet eligible. We wanted them to be around for at least 5 years. Do you have the ability to provide usage data, quantitative usage data on the usage of your website? Do you use metadata and the appropriate standards? I mentioned that. Is the data available? What's the format? Do you have appropriate governance in place? For instance, is there a scientific advisory board or a scientific advisory committee? And do you have an open data policy? And the answer to that was, we didn't want them to say yes to that. We wanted them to be able to cite a specific policy that was shown on the website that described the availability of the data. Okay. We expected a yes answer from all of these in order to proceed to the next round of applications. In some cases, the respondents gave somewhat vague answers. And we allowed them to proceed to the next round, but we also said you must clarify this in order to be evaluated in the next round. This was particularly true of the open data policy. A lot of them said, yes, we have an open data policy, but we couldn't find this written down anywhere. And we asked them to write it down and make sure it was on their website. And most of them did, because they actually had the policy. They just hadn't put it on their website. Eligibility criteria, here's a couple of examples. Can you provide statistics? Do you have a scientific advisory board? And do you have a data license that demonstrates that the data are open? There were some short answer questions, and these were really the meat of the application. So there were just a few questions here. That asked about the alignment of the data resource with the goals of the GBC and the characteristics of the resource. And the idea behind these was to understand if this resource had a realistic chance of being named as a global core biodata resource. And here's a few examples of those questions. And these are short. We usually limited them to 200 words. Short as almost essays, just a few sentences. How does the resource serve the scientific community? How many users do you have? How do you support your users? Do you have a help desk? Do you do training? And do you have the appropriate management structure and governance in place to be a global resource? So importantly here, a global resource should be one that has a little bit more formality in the way it operates. We want them to have a scientific advisory board. We want them to have a defined management structure. It's not just the professor in the lab and one of the graduate students running this from their laptop in the office, okay? So as I said, the review committee evaluated these expressions of interest, which were fairly short, fairly easy for the review committee to read, fairly easy for the applicants to write. And then the review committee decided which ones had a chance or were likely to be able to be named as global core biodata resources and pass them to the next level. And the image here is the software we used to process the applications. Now we purchased a license for this software, which is called SurveyMonkey Apply, and it's a software that's designed for granting agencies to allow grant applications, and it was quite appropriate for this, and it's quite professional. The applicants have access to their application, but the reviewers have separate access to be able to review it. There's a separate portal for the reviewers to submit the reviews. So it's very took a lot of time to set up, but once it was set up, it was very straightforward for both the applicants and the reviewers and us as managers to manage the applications. Sends out automatic emails. It does a lot of things which made it much easier to run the process. So you don't want to do this with an Excel spreadsheet again from an office somewhere. You want to do this, set this up professionally. It takes a lot of time at the beginning. It was fairly expensive, although there are less expensive versions of software. There is less expensive software around, but in the end, it pays off in the professionalism and the efficiency of the process. So I'm going to go through just a few of the longer questions that were on the full application. All of them were expansions of the shorter questions on the expression of interest. So I'm not going to read this, but we just wanted the data resource to describe its scope. What is it doing? What kind of data does it have? And which research community, even if it's everybody, is it serving? And what kind of data resource is it? So here's an example of a quantitative number, quantitative usage. So how many we want them to distribute. We want the resources to demonstrate that they're used extensively, both in terms of the number of users and the distribution of their users. So if I live in Cambridge in the UK. If the resource is only used by people in Cambridge, that's not a global resource. Could be very important in Cambridge, but it's not a global resource. And we've asked them to actually, in the expression of interest, we said, can you provide this information? In the full application, we said, now give us the information. How many users do you have? Usually that's not really how many users, but how many IP addresses are accessing it, because that's the number that we can get. Here's an example of what that looks like, the quantitative data. How many visits per month, how many unique IP addresses, how many page views per month. And there's different ways of reporting this. There's different kinds of software that records it. So we give them options to report to us how the data was collected, how the information was collected. And we wanted 3 years of information from them, the past 3 years. And we made a provision for some complicated situations, which is something about what that mentions. Concatenation thing is at the bottom right, but I won't talk about that. Now, an important thing to remember here, though, is that even though this is quantitative data, the reviewers cannot take this information and just line it up, because the number of visits per month, the number of unique IP addresses, the number of page views, that can vary tremendously for different resources. But those resources could be equally important. And different numbers could be large or small for different resources. So one number might have a lot of unique IP addresses, but not many page views, because each user comes in and looks at one page. But one resource could have not too many IP addresses, but many page views, because each user who goes all over the place and looks at many pages. So there's different ways of using the resources. And this contributes to the difficulty of evaluating them, and it's a challenge to the reviewers. So again, then we asked them, how many times has this resource been cited in the scientific literature? And when we asked them to report how they were figuring out that number. Some resources have accession numbers for the submission for the data in the data resource. We asked them to, if that was the case, we asked them to report how many times those accession numbers have been found in the literature. Does the resource have a scientific advisory board? We said, again, I said this is very important. So we asked them this. How open is the resource? So this refers to the licensing again. Does it have a license? Does it have an open license? And is that open license stated clearly? So the conclusion here is that if you're going to do something like this, it's really important to base the work on what has previously been done. So obviously everyone is familiar with grant applications. We really base this system on a grant, the operational aspects of this on the grant application system. The criteria we used were actually based upon previous work by Elixir, which is an infrastructure in Europe for bioinformatics and biodata resources, who developed a process for identifying Elixir core data resources in Europe, when those are resources only in Europe. Now, I have to confess here that at the time I was working at the European Bioinformatics Institute, and you'll see my name on there. So I was also involved in developing the Elixir methodology. So there was some connection of people here, as well as just a method. So I think that some of the more senior scientists in this room and elsewhere that we've talked to over the past week are working towards developing a more defined an organized bioinformatics community in India, so that all of the researchers in India who are dealing with data resources and bioinformatics can work together to create a cohesive and connected infrastructure, much as Elixir is in Europe. So one thing that might be done there is to develop a method to identify resources, data resources in India that are important within the Indian context. And I certainly hope that's done, because it would be an interesting thing to do. There are probably thousands of resources even in India. We counted 3,000 resources, but we know that was a gross undercount, because why we know that is described in our paper. The message here is that we'd be happy to provide advice in helping to adapt this method to local use and for local requirements, and that will mean going back to those indicators that I just showed and getting rid of some of them, maybe adding a few more and tweaking them a bit. But the other key aspect here is to have independent reviewers who are not connected to the funders, who are not connected to the whatever group is running the selection scheme. So that's all I have. Welcome. Maybe one question if we have time. Otherwise, questions to the Google Doc.
Yeah, we can have one question if somebody has a very pressing question.
I couldn't hear you. That's a really good question. We did one in 2022. We did one in 2023, and there are now 52 resources. But we're not going to do it annually, because the first time, of course, we got all the resources. It's never been done before, so there was a lot of interest. We don't expect that the infrastructure is growing that much so that we'd want to do it every year, but it's hugely time consuming. There are categories of resources that we did not allow to apply. So for instance, there are open data resources which have human data, but they're open in the sense that the data are open, but the users still have to request access permission. We did not allow those kinds of resources to apply this time, but we should, we will in future. We just haven't developed. We have to expand the method, the selection process, to understand how to do that. So the next time we do this, 2 or 3 years from now, we hope to expand it to include those kinds of resources.
Yeah, so we thank Chuck for wonderful talk. After learning about the Herculean efforts of global biodata collation, it's now time to learn more about what's exactly happening in India. And in fact, the title of the symposium was motivated by our next speaker's presentation title, which was Life Science Data Revolution in India. So I now invite Professor Saurabh Raghuvanshi for his talk.
Thank you. So I'll start by thanking Professor Ashwani for giving us this opportunity to present. And I won't be taking, this is not a very long lecture. What I want to do here is to discuss how perspective has changed about big data and how it has to move on. So, yeah. So I would be taking examples from something which I have been involved deeply. And the first slide probably is to take a perspective, to take an idea of the perspective, how our thinking as scientists have evolved about data. Now, this is something that happened about 20 years back. This was probably the first project where India indulged into a big data generation project. This was the Rice Genome Sequencing project. And at that point of time, you can see over here that India participated in sequencing half of the chromosome 11. So this is the time when bag-based and map-based sequencing was done. And this was the time when 1 terabyte of data, the disk box that contained 1 terabyte of data, weighed about 10 kgs. Now that 1 terabyte of data, probably you can have it in your cell phones. So that's the amount of change that has happened. Now let's see how the perspective has changed globally. Now, if you imagine how databases developed, most of the repositories or most of the curated databases were basically model systems that were being worked upon. Either it were humans, mice, or Arabidopsis and rice. So that shift has happened at a global level where people are not just looking at model systems, but they are now looking at entire biodiversity over here. And this basically means that projects like Earth Biogenome, which actually targets to sequence all species, that's a change in perspective. Now, what does that entail? What does that mean? One of the most important thing that happens is that there is an absence of benefit sharing obligations. Now, keep this word in your thoughts and we'll come back later on this slide. Now, another aspect that another change in perspective. When we are saying that we are shifting from model systems to entire biodiversity, so biodiversity is everything. But then what about crops? So our database generation as well as curation of the data, that perspective has also changed, and it has to change a lot because now we have to generate data on crop systems also, not just model systems. And this phenomena actually is going to be specific to a country. For example, millets. Now, millets is something that we want are consumed over here, may not be consumed in Europe to that extent. So this is something that India has to do on its own to develop databases and curate information on these aspects. So the concept that you can keep on working on model systems and do all the biotechnology based on that, that has significantly changed. Another important aspect that has changed is that earlier, the genome, things like genome sequencing or things like the structural biology, especially genome sequencing or NGS data, this was restricted to genome labs. So they were pretty accustomed to on handling high throughput data sets. But now, as we know that the high throughput data is actually entering into every researcher's lab. We are having RNA-seq data in every lab, we are having chip-seq data. So that's a change in perspective, in the sense that now every lab needs to be proficient in managing the data. Now, we are talking about data, then what is the big data? So classically in computer science, big data is defined by several V's. So these are the 3 V's. There are several more V's actually as things evolve. So it is the volume of the data and the velocity with which it is created as well as the variability. Now, another important aspect that has happened with big data when it starts to accumulate is there's a difference between data and knowledge. It's a big gap. So generating RNA-seq data doesn't mean that you have an information about what is happening. You need to analyze, you need to integrate a lot of data sets into that. So how does that happen? This is basically another dimension of perspectives, in the sense that when you take a RNA-seq data or any high throughput data, for example, if you generate any high throughput data, you would generate it with a particular perspective that I'm looking for this stress and that is something I want to work on. But that is not the entire information that data sets have. That basically means that this big data, which is generated by any lab, needs to be analyzed by several different perspectives to actually generate the entire knowledge that data set provides. So that basically means that the data needs to be available in a very fair fashion. Now, FAIR is an acronym here, which stands for, F stands for findable, accessible, interoperable, and reusable. Now, if we just give a moment to how human genome data was, what is the economic or social impact of the human genome data? It is huge in billions and billions of dollars. But how did that happen? Simply because that the data was made fairly accessible to everyone on the globe. But there's a debate, and I'll tell you what the debate is, and then I'll just summarize why that is happening. The debate is that there is a tug of war between this thought that whether the data should be made totally open access all the time, or it has to be a restricted access. Now, the point is why people are saying about restricted access, and this is because, as I said, access benefit sharing mechanism on biodiversity. Now, what is this? Now, this is a convention on biological diversity. So this basically was a convention on conserving the biological diversity across all nations. And the idea was that if you have some economic benefit from that biodiversity, that benefit has to go back to the community from which the data was there. So there's a term over here which is known as genetic resource. And this term genetic resource was basically used to define a live tissue. That is, if you move a live tissue from one country to another country, we're not free to do that kind of a thing. But now what has happened is that this definition has now been extended to include even the DNA sequence which is coming from that information. So that is known as DSI, that is the digital sequence information. So now that basically means that if there was a restriction on the movement of the tissue, then there should also be a restriction on the movement of the data. Now, why people are saying this? So this is a very simple cartoon to say that. So if you have a live tissue and you are commercializing it, you can restrict the movement of the tissue. And then you can say, okay, whatever benefits are there, they will be shared back to the community. But see what happens now. Because the data that is generated over here is open access, you actually don't need any tissue to generate any metabolic pathway. You can have actually manufacture or engineer any metabolic pathway if you have the genome information of that. So basically that means that this is being perceived as an infringement of this phenomenon. So there's a tussle. It's not a set, it's not a done deal. So nations are actually debating on whether DSI, that is the digital sequence information, should be under a similar kind of a lock as the movement of the tissue. Now, keeping that thought, again, we'll come back on a later slide. One of the major impacts on data analysis, on impact of data science, at least in India and for all nations, is the national food security. And it stands on three pillars, actually. One is the gene to trait association. We saw that speed breeding thing, that's wonderful facility. That's a gene to trait association. Then you basically have a smart breeding. I won't go into the detail of a smart breeding. There's a lecture on its own. And then precision agriculture. And if you see, all of these are based on basic data sciences. As things have moved, data science has become actually fundamental to all of these. Now, to answer these questions and to address these questions at national level, the data center had come up where I was working a few months back. The basic idea of this data center is to actually have archive, curate, and dissipate all the information that is being generated from India, and then have a lot of international collaborations. Now, when we talk about data center, what is it that we are talking about? It's very important to understand that it's not just infrastructure. It has to have inclusivity. It has to have internationally accepted data submission and sharing formats. It needs to have a very clear-cut and transparent data policies, data security and clear-cut confidentiality assurance, dedicated staff, and most of all, the right attitude. The right attitude in the sense that the data center staff needs to understand that it is working for the entire scientific community. So it's a service. It's not just keeping a server at a premises. So this is what happened. I joined in March 2021. In 2-3 months, we were able to launch the portal, and we started pushing off the open access data to the European Nucleotide Archive. And within probably 1 year, we were able to generate, we were able to accumulate more than 100,000 sequences. And one of the most important things that has happened was that it was not just data deposition services. We were able to actually assist the nation in several very important projects. One of them, the first that we picked up, was the INSACOG, because this, if you recall, was the time of a total lockdown because of the pandemic. So the data center actually provided the services for data deposition as well as data analysis for the INSACOG, which actually is a consortium of about 40 to 50 hospitals, research institutions across India. And the news that you see that this particular strain is now most prevalent is actually coming from the analysis performed at the data center. But you may not see the name of the data center. So that basically means the data center is actually facilitating all these major projects for their work. Another major important event that you must have seen recently was launch of the 10,000 Human Genome Project in India. That data also is being archived at the data center. So it is not just data deposition, it is to enable such large projects which are generating data set. That's the basic mandate over here. So this is the facility. You're most welcome to visit this. I won't take much time over here. Now, what is this? This is basically, as I said, it is accumulating data. Now you can't have a single portal which accumulates all kind of data. So there are several portals. These two are INDA and INDACA are the Indian Nucleotide Data Archive. And this is control access. This is ICPD, that is a phenomic data sets, plant crop phenome data sets. These two are operational. These three are operational. Now, what happens is, for example, for metabolomic data and proteome data and image, the development of these portals are under process, and they go through these different stages where the development of the portal is actually done in sync with the scientific community, both at national and international level. And since we are developing now, that is, as compared to other international repositories, we are developing these portals within a single data center. There's not different data centers working for these. So all single data center with all these data types. So we're keeping the data types. This is slightly complicated, but what you need to understand is that whenever you take a data set, any kind, you add a lot of metadata to it. And the way it is added is very important for its search, because you won't be able to search the data if you don't add your name to it. So that metadata is kept very constant, so that all of these data sets for a particular gene, for a particular species, for a crop, they are all integrated in a fashion. Now, something that is very central to this entire thing is a community-wise realization that the aspect of data generation and knowledge is bridged by something what is known as biocuration, that is integration of data and adding metadata. So we have been trying to advance this line of activity in India. We have been performing, we have done several symposiums earlier, and later on we ended up doing this conference just last week. It went very nicely. We had participations from 15 different countries, and over 100 participants, 100 research institutes were actually represented over here. So it was a great success. We had a workshop at Delhi University South Campus also, where we had the opportunity to have data heads of European Nucleotide Archive, Professor Dr. Guy Cochran, Professor Masanori Arita, who is heading the DDBJ, and Dr. Eileen Mizrachi, who is heading the NCBI Sequence Archive, and also Professor Kyoko on glycobiology. So the basic idea over here is that the perspective with which every biologist must perceive the data has changed a lot. It is not something that remains with the bioinformatician. Bioinformatics is something like molecular biology, which is actually for every biologist to learn. This is, I must acknowledge my group. So this, actually the work of this group is the South Campus group, the work I did not discuss today. This is the work at database, which I discussed today. And yeah, thank you.
Thank you so much, Professor Raghuvanshi. Maybe we can have one live question, and then anybody? Yeah, it's really fantastic. So to run this IBDC, a lot of resources are required, and like yeah, exactly. Yeah, and it was really interesting to learn about this global collation which is there for this biological data. So I once again thank you very much, and please stay here, and I invite all the other remaining speakers to join on the stage, please. And also to Executive Director. Yeah, please have a seat. So let's give a very big round of applause to all our speakers once again. Thank you. And I now request Executive Director Professor BRIC to please present memento to all our speakers. First, Professor Guy. Yeah. Yeah. Yeah, first Professor Guy. Yeah. Yeah. Thank you. Professor Chuck Cook. Thank you. Yeah, next, Professor Saurabh Raghuvanshi. Yeah, now it's my pleasant duty to propose vote of thanks. First of all, thanks to all our speakers, then to my fellow colleagues who helped me organize this event, and also to my lab members, and also to our admin staff, and also to our executive director for his initiative to organize this event and his guidance. And thank you everyone once again. And now I invite you for refreshments. Have a good day. Thank you.